Assalamu alaikum. You're watching Microbiology by Nouman. In this video, we'll talk about a very important concept of microbiology, which is known as hemolysis on blood agar plate. We will also discuss what is beta hemolysis, alpha hemolysis, and gamma hemolysis with their examples and pictures. Before we start this video, please like this video and subscribe my channel. So let's start. So the first part of this topic is hemolysis. Hemolysis means lysis or breakdown of red blood cells. Red blood cells are also known as erythrocytes. So hemolysis is lyse, and the second part is a blood agar plate. In microbiology, we use a different type of culture media to grow bacteria and other microbes. So one of the example of most commonly used culture media is a blood agar plate. So blood agar is a culture media. It is made up of tryptic soy agar and contain around 5% sheep blood. This blood part make this blood agar plate a very nutritious for bacteria to grow. It allows many bacteria to grow, not all, but many bacteria can be grown on blood agar plate. So breakdown of RBCs on the surface of blood agar plate is known as hemolysis on blood agar plate. Blood agar plate is also written as BAP, blood agar plate, right? There are certain group of bacteria that can completely break down RBCs, and there are certain bacteria that can partially or cause incomplete hemolysis of red blood cells, and their third possibility is a bacteria that do not cause any kind of hemolysis. On the base of degree of hemolysis, we categorize hemolysis into three categories. The first category is beta hemolysis. Second category is alpha hemolysis, and the third category is gamma hemolysis. First, we'll talk about beta hemolysis. So, beta hemolysis is complete breakdown of red blood cells or complete lysis of RBCs on the surface of blood agar plate. So, complete hemolysis and complete lysis of RBCs. This beta hemolysis is due to bacteria produces enzyme known as hemolysins. Example of hemolysins are streptolysin O and streptolysin S that are produced by streptococcus pyogenes. There are other hemolysins as well, right? So if we look at the bacteria that causes beta hemolysis, if this is a blood agar plate, for example, and bacterial colonies are visible on agar. This is a blood agar plate. It's red in color, right? Because it contains blood, so it is red in color. Without blood, tryptic soy agar is a transparent or yellowish color agar, right? Around a bacterial colony on the blood agar plate, we see a bacteria that produces beta hemolysis. There will be a zone, clear zone or transparent zone. Around a bacterial colony, there will be transparent or clear zone. Or clear zone. Zone means area around bacterial growth. if we'll I will paste an images of different type of different bacteria that produces beta hemolysis. A very common example of bacteria that produces beta hemolysis is Staphylococcus aureus. Staphylococcus aureus produces golden colored colonies on blood agar plate, and around the Staphylococcus aureus plate there will be a clear zone of hemolysis which is known as beta hemolysis. That zone will be transparent or yellowish in color, right? Example of beta hemolytic bacteria are Staph aureus or Staphylococcus aureus, Streptococcus pyogenes, Streptococcus pyogenes, and Streptococcus agalactiae. If we look at this image that is taken from the American Society of Microbiology, there is a blood agar plate that contain only Streptococcus pyogenes. Around the Streptococcus pyogenes, a clear visible zone or transparent zone is seen around Streptococcus pyogenes. After a few days, a plate of Streptococcus pyogenes becomes so transparent that if we put blood agar plate of Streptococcus pyogenes on the surface of a newspaper or any book, we can easily see or we can easily see across the transparent media and read the newspaper as well. So beta hemolysis is complete hemolysis of red blood cells. It is caused by hemolysins, and around the bacteria there will be clear zone which appear as transparent or yellowish in color due to the streptococcus agar. An example are Staphylococcus aureus, Staphylococcus pyogenes, and Streptococcus agalactiae. The second category of hemolysis is known as alpha hemolysis. There are certain bacteria that cannot completely break down RBCs. So when they break down RBCs, resulting product is an incomplete hemolysis, right? So alpha hemolysis is incomplete or partial hemolysis. This alpha hemolysis is due to a bacteria produces hydrogen peroxide that will convert hemoglobin into methemoglobin, right? Resulting in a greenish product, which is methemoglobin, right? So if you look at the plate or containing a alpha hemolytic bacteria growing, if this is a blood agar plate, right, and this is bacterial growth, around the bacteria that produces alpha hemolysis, there will be a greenish zone, right? A green zone will be visible around a bacterial colony, right? So greenish zone is visible, and that indicates the alpha hemolysis. Due to this greenish product, this alpha hemolysis is also known as green hemolysis or greenish hemolysis. Example of bacteria that produces alpha hemolysis is Streptococcus pneumoniae. Another example is Streptococcus viridans or viridans streptococci. If we look at the picture that I pasted here of the alpha hemolytic, we see around the bacterial colony a green zone is very visible that indicate the alpha hemolysis. So alpha hemolysis is incomplete or partial hemolysis of RBCs or red blood cells or erythrocytes, whereas beta hemolysis was the complete hemolysis of RBCs and resulting is a transparent or clear zone, right? Whereas in alpha hemolysis, greenish product or green area around the bacterial colony can be seen. The third category of hemolysis is gamma hemolysis. There are certain bacteria that do not cause any kind of hemolysis or breakdown of RBCs. Such a hemolysis is known as gamma hemolysis. So, no hemolysis of red blood cells on blood agar plate. Remember, these kind of hemolysis can only be seen on the blood agar plate that is containing a 5% sheep blood, right? So alpha hemolysis is no hemolysis. If we look at a blood agar plate that containing a growth of alpha hemolytic bacteria, blood agar plate is red in color. After this growth of bacteria in the form of colonies on this plate, there will be no change in color, and around the bacterial colony there will be no color change, and blood agar plate will still appear as red in color. Example of bacteria that produces gamma hemolysis is Enterococcus faecium. Another example is Enterococcus faecalis. Other examples can be Staphylococcus epidermidis and Staph saprophyticus. Epidermidis and Staph saprophyticus. So in short, beta hemolysis is complete hemolysis and resulting in a clear or transparent zone. Alpha hemolysis is incomplete or partial hemolysis of red blood cells on the blood agar plate, resulting in a green discoloration around a bacterial colony. And gamma hemolysis is no hemolysis or breakdown of red blood cells on blood agar plate, and there will be no change in color of the agar due to these bacterial colonies. This alpha, beta, and gamma hemolysis is very important in streptococcal classification. Their streptococcus are gram-positive cocci bacteria that are divided into different groups on the basis of their hemolysis pattern. We'll talk about it in the next video. For more video, you please like, share, and subscribe my channel. If you have any question regarding this topic, you can ask in the comment section. Thank you.